imaging, and it's pretty well equipped for doing multidisciplinary work. And we are funded by extensively by various agencies, including multiple NIH grants and other federal agencies. This is my group. It's kind of comprised of multidisciplinary skill sets from chemistry, from materials to biology, and engineering students, including MD, PhD students. There are three research areas mainly we cover. It's mostly on point of care biosensing, therapy, drug delivery, and also one of the earliest themes that we started with was imaging and in developing probes for molecular detection by using various clinical and preclinical imaging modalities. So one example that I wanted to give here is a molecular imaging. So you know that this is probably familiar with this image. This is December 22nd, 1895. This is the first X-ray image, black and white, everything, no soft tissue contrast was seen. And you can see here that this is the gold ring that was seen, and this is William Ronsen's wife's left hand. This is a very famous X-ray image. And now, if you look at fast forward, you can see that the CT imaging in that has improved quite a bit, but we still do not really see any soft tissue details, and that we want to change that. That how can we really bring more clarity to these images, and how can we really detect things at a molecular level? So this is an example of our lab, and that's showing that this is a human bone, and we were targeting multiple proteins at a time and enzymes. And this is an example of an osteoarthritis that shows that how different protein targets can be proteins can be targeted by using nanoparticles and be imaged by emerging techniques, for example, photon counting, computed tomographic imaging. So remarkable clarity and resolution with very high soft tissue details can also be seen, and with molecular information with these contrast probe. And here also you can see that this is an example of human bone, where you can see this is a screw, or this is for after the surgery that has been implanted, so we can clearly see it by and separate as a different color. So we can generate color images, and this is also our lab's example where we're seeing that this is a bone and this is a micro crack where the nanoparticle is actually targeting and latching onto the micro crack. So if you image this by a regular X-ray, you won't be able to see the micro crack, but now when you're targeting it with nanoparticles, you're able to see it very quickly, and then image it with photon counting CT. So the photon counting CT is a newer generation of CT, and this is not really the topic of today's discussion, but very quickly that in regular CT, we have an energy integrating detector. It does not have the capability of counting photons, but in photon counting CT, we're now working with the semiconducting detectors, and they have the capability to count each and individual photons that is coming through the X-ray tube. And depending on the bandwidth of the X-ray and the K-edge energy of the element that we are using, that we're interested in, the system can generate a signature peak. For example, gadolinium here, it's right in the middle. It's about a 50 keV of K-edge energy. Gold here, it's about 80 keV for a K-edge energy. So they're going to generate a very strong signal, a signature peak that is highly specific to that particular metal. So this is a quick concept that is showing that there are different energy bins, and we can put these nanoparticles under different energy bins, and in theory, we can generate a very colorful images and precisely telling like where exactly these metals are going and how these metals are going to show up in a body. So this is an absolutely brand new area of research. Now, as you know, that nanoparticles, and this is again, this is a center for nanomedicine, so we all are familiar that nanoparticles, once they go in, they must come out. And for inorganic nanoparticle or crystalline nanoparticles, and that is even true. That is even more applicable because the inorganic nanoparticles or crystalline nanoparticles, they do not have an ability to get digested inside the body. So for inorganic particles, the particles must be less than 10 nanometer in size because the kidney cutoff is 6 to 8 nanometer. So that's one of the reasons why we are interested in particles that are very tiny and that can produce a signal. Also, with because of their high crystal packing of the atomic crystal packing in these particles. So these are some earlier works that shows that we can do a whole lot of studies. For example, we can detect fibrin protein that is present in blood clot, and you can see that this is an example where we're showing that this is a human a carotid endarterectomy specimen taken out from a human after the surgery, and we are now incubating it with the nanoparticles, and you see the nanoparticles are now binding fibrin present in this tissue, and this is a calcium that can be clearly separated as because the both these these are bismuth nanoparticle. The both these bismuth and calcium, they have different K age energies. So here one example that we very recently we prepared, making these particles in a reproducible manner for and tiny particles. So these are ytterbium particle and also gadolinium we made, and you can see that these are about 5 nanometer, 6 nanometer in size. And beautiful crystal packing here. You can see from the more crystal packing, and this is an example that we are now delivering these particles by pulmonary delivery. So this is a pig. Sorry, this is a pig study that we are doing here. This is in collaboration with our collaborator in France. You're seeing that how the pig is. We're generating and these are the particles, and you can see that the nebulization is happening, and the aerosol is being formed, and it is being delivered in the pig lung. And so this is the first demonstration of a pulmonary delivery of nanoparticles in pigs. And you can see that how this is immediately after the delivery. You can see that the particles, this is the purple color is actually it's a nanoparticle, and these are rib cages. You can see these are bone. This is 30 minutes after delivery. You can see that the particles are now coming in the apical region of the animal. So again, this is a beautiful image that shows, and this is a video that shows that how the CT imaging works. You can see clearly where the nanoparticles are located at, and the particles are and now very clearly seen with no ambiguity with the surrounding of the bone and the lung region. So this is one of the reasons that and also we can do multiplexing imaging in vivo using this color CT, because this is not really possible with any other clinically relevant modality. So for example, this is a we published in Advanced Science last year. It shows that here you can see that there two targets. We're targeting two markers here. And then one is agrican and the other one is agricanes. And these are both are related to osteoarthritis. So this was done in osteoarthritis mice. And we're showing that how these the two particles we're using, and you can see the relative expression of these particles, and they are completely quantifiable. So you can actually know that where the disease is and how much the progression of the disease happened. So two biomarkers we can target of interest in vivo. Control delivery, we're not going to really talk about it, but I think we're going to focus a little bit on innovation in devices and point of care sensors. And you know, my lab has been developing sensors for quite a while, and we are known for our work in the regeneratable electrochemical sensor. And our patented technology shows that we can detect genomic targets in pathogens. And the sensing principle is based on what we call a binding induced folding sensing, where we have a working electrode, and typically this is a gold or carbon electrode, and then we have an antisense oligos or peptide nucleic acid that are placed on the surface of these electrodes, and these are typically forming a hairpin loop structure. Now imagine that a target RNA comes in and then and finds the complementary binding region. When it binds, then this hairpin loop structure is now kind of becomes linear. Now it is straightened out, and the electron transfer is now stopped. And so we're using methylene blue as a redox reporter, and this is an amplification free approach where the capture probe is unmodified. Only the signaling probe is modified with the redox reporter, and it has been applied for looking at a single base mismatch information. So extensively studied for SARS-CoV-2 and for other pathogens. And here one example that we were showing that we can look at SARS-CoV-2 and the variants of their concern, and we can precisely go up to a single point mutation. You know, that is basically a consecutive of three nucleotide sequences at the target side, and only can reduce have activity in case of there is a dual or triple point mutations. So uniquely identifies the SARS-CoV-2. You can see that this is kind of a little bit of an old data, but it shows that how precisely it detects with about 98% sensitivity and specificity and accuracy. This can be seen. These are all patient samples that you can see that the positive predictive value and negative predictive value for these sensors. Now, we're constantly looking for developing new things. So, for example, here in the 2D nanomaterial field, you know that graphene has dominated quite a bit, right? In the for over 20 years, and you know, however, you know, the expectations about its impact was what we now realize that overestimated. So, because my lab also worked on graphene. So, for example, about 14 years back, we developed a graphene-based platform, and that was the basis of starting one of the first sensing-based companies from my lab, which is still this company is active on in developing a sensor for ocular diseases. But we realized that the graphene is not really the best material for translation. And as a result, the interest in exploring other zines that really happened, that really there's been a huge surge in developing things. For example, MaxZines and other type of things, including borophene. So boron and borophene is was discovered in 2015 around, and it's very interesting material. It can exist in various polymorphic forms. It's a two-dimensional allotrope of boron that was kind of theoretically predicted around in 1990s, but then actually synthesized in 2015. It's a atomic crystalline atomic monolayer of boron that forms a metallic structure with mixed triangular and hexagonal patterns. So very interesting in terms of its structure. Now, is it really a wonder material? Like, for example, what kind of properties that make and set borofin apart from other materials? It has an outstanding electronic mobility, and that is comparable to graphene and even better along its ridge lines. And in contrast to other existing 2D materials, for example, phosphorine and TMDs, it has a much better electronic mobility. It is metallic, and in contrast to semi-metallic graphene, it's completely metallic, and the thermal conductivity of borofin is just below that of graphene and boron nitride, however, better than all the other existing 2D material. It exhibits a Young modulus that is higher than graphene along its ridge line. So you can realize that this is an amazing structure that is a very spatial structure that has a negative Poisson ratio, and that makes the atomic structure as auxetic. That means it can be used for potential application in flexible electronics and wearables and biosensing. So our interest kind of started from there, but it's not really easy to prepare borofin. So we developed a top-down liquid phase exfoliation procedure from a boron powder, and we were able to get to a Chi3 boron phase and beta 12 boron phase. And you can see here, these are some beautiful TEM images that you can see. But the problem with borofin is that their surface is highly reactive and it's prone to aerial oxidation. Surface oxidation and functionalization and with passivation with agents like proteins and other type of things that can help stabilize these materials. So here's some characterization, and this work was published in I believe in 2025 in ACS Nano. And you can see that how these Chi3 borofins are presented here, and this is the beta 12 phase. Also, we can change the condition a little bit. Intermittent sonication in cold condition will lead to a majority of the beta 12 phase. But then what we showed that we can put amino acids on the surface of this borofin material, and what we prepared was a chiral borofin by doing a site selective chiral induction. So here you can see that how we are really putting very simple room to room temperature process for 12 hours. We are putting amino acids on the surface of these materials that render borofin chiral, but at the same time it also makes them chemically stable and much more usable. So one of the things that we recently did is used borofin in lateral flow assay. So here you can see that how we are using. So this is a borofin ACM. This is a nitrocellulose membrane that are typically used for lateral flow test strips. And this is borofin and nitrocellulose material. You can see the borofin plates are clearly seen here. So this is old slide, and this was published in ACS Central Science last year. And then how we are really putting the antibodies is really interesting, because what we wanted to do, that we wanted to address a key issue in biosensing by a biofunctionalization of the active surface, since that kind of deeply affects both the sensitivity and the specificity. So the antibodies, what we were doing, that we were taking the antibody, and then by using some UV and photo irradiation, we are irradiating the antibody, and that generates thiol. And so when the disulfide bond breaks, the thiol is generated, and that will be anchored on the surface of the borofin. The way it does that it's really the one fragment fragment or antigen binding fragment will be exposed to the surrounding environment, and the other fragment will be on the surface of these borofin material. So it's a site selective immobilization of antibodies on the borofin surface, and that gives a high specificity, and that in turn leads to a superior sensitivity for borofin. So here you can see that this is an antibody borofin gold sandwiched complex, and this is a transmission electron microscope. I'm not quite sure whether you can appreciate that these are gold particles, and this is and in the background you can see that there are borofin, because borofin is a very little contrast. You cannot really see borofin that much, but in gold you can clearly see the gold particle. But this is HADX, and you can see, or HADF, you can see that the borofin is in the background in red. In green, you can see that the gold particles are clearly seen. So these superstructures are formed by the interaction of the Fab region of antibodies with gold nanoparticles on the borofin. So this is a morphological analysis using SEM. You can see this is no borofin here, and this is with borofin. You can see that how the particles are exposed, and you can see a lot more particles present on these nitrocellulose membrane surface. We studied extensively their isothermal titration calorimeter, ITC. There's not time to go over these things, but here one example that we did for human menstrual effluent or period blood, and we detected a protein biomarker, and that's a target protein HMGB1, and that is linked to endometriosis. And what we are showing here is that this is a conventional LFA with no borofin, and you can see that we can detect very easily up to 250 a picogram per ml. But then this is an improved LFA with borofin. You can see we can detect it up to 50 picogram per ml. So this is remarkable sensitivity, and you can see the linear dose response curve for both conventional LFIA and borofin conjugated or borofin enabled LFIA. Can we decorate borofin with DNA? Yes, we can decorate, and these are all unpublished work right now. But we showed that we can actually put DNA on the surface of borofin, and this process is really unique because the ASO, the antisense oligos, they can take two conformation, and one is actually a vertical conformation, and the other is actually a horizontal conformation. We did extensive ITC studies of nucleobases and oligos with borofin, and we also did atomic force microscopic studies to understand that there are some hydrophobic pockets inside borofin, and that that kind of triggers this bimodal interaction, a very two well-defined interaction. One is a thiol interaction that is predominant as a result. We see for as an increased height at a 3 nanometer, whereas the hydrophobic pockets are showing a less increase, and that is a 1 nanometer in height, that we are seeing that's a little bit less. Here you can see that that a secondary orientation is also seen. So these are all very, 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 very unknown right now. So fundamental studies. But again, I think keeping in mind that we would really like to use these things for real world applications. What we did that we use this borofin surface as a nano surface energy transfer application on borofin, and what we are finding, so this is a fluorophore tagged with HIV antisense oligonucleotide, and now we are looking at the quenching of fluorescence with borofin nanosheets, and the regain of the fluorescence signal with addition of the target gene was clearly seen, and initial results show that almost 50% of regain of fluorescence. Here you can see that this is a HIV-1 plasma samples, and the LOD was 37 copies per microliter, and with a very high accuracy and specificity. I'm not going to cover this. I think one thing that I wanted to say that borofin is completely biodegradable. Our recent work is showing that borofin can be completely biodegraded, and once they're inside the cell, we also know how they're going inside the cell and what kind of pathways they are taking. So there is this is a video of a holotomographic image that shows that how borofin is actually, if you can see that this is borofin, the black dot here, it is going inside the cell of A549 cells, and this is a real-time image or video showing that how the particle is actually going inside the cell. And this is a refractive index based technique in 3D holotomography that is showing that how these particles are being taken up by macropinocytosis as a predominant a mechanism for taking it up. And these particles are actually completely degradable in inside the cell. In 24 hours to 48 hours, these particles are completely gone. With that, I think I'm just going to say that borofin's potential for biological application is really untapped because we're still trying to understand how these particles can be made in a reproducible manner and in a scalable manner. Excuse me. So we developed a process that is highly reproducible and top-down with liquid exfoliation that makes a different polymorphs of borofin. Um, We developed various processes for strategies for surface passivation using amino acids, nucleobases, antibodies, and DNAs. Um, our results indicated that these materials can be successfully used for protein, nucleic acid, and sensing in complex biological fluid. Deeper understanding of their interaction with biological system is needed, and that's what we are right now. We are working on. This is my team acknowledging them, and finally, thank you so very much again for your kind invitation. And I'll be happy to answer any questions that you may have.
Thank you, Dr. Pan, for your scientific deliberation and interesting insights about the nanoparticles and their biosensing applications. The session is now open for the question and answer. Yeah, Karan. We have the mic, please. Um,
Hello, Dr. Pan. My name is Karan, and I have a question for you. So, do you think that the multi abilities platform, nano platforms that you are making, would face rigorous regulations from agencies like FDA than the currently available single purpose materials that are there commercially?
You mean so you're asking about the imaging application?
Yes, sir. Yeah, exactly.
Yeah, sure, absolutely. Yeah, there is no doubt about it, and that's one of the reasons why we want to make it a slightly different, right? We don't want to do an intravenous injection and a systemic delivery. We want to do that by using a pulmonary delivery. So that's a um, that's an approach that we are taking, so that it does not really see the key challenge for FDA is that it gets to the blood and it's taken up by the macrophages and then it gets cleared by kidney, sorry, liver and other reticular endothelial systems, right? So that's a big challenge, and that is a main challenge for nanomedicine community. Um, Multiplexing approach is actually one of it's I would say this is a less clinical approach but more as a fundamental discovery to show that the particles can be used for a molecular detection of multiple proteins by using a clinical modality which has not been done so far. But to answer your question, yes, absolutely, this will this will have to go through the same FDA regulation process and so on. The question is whether the benefit outweighs the advantage of using two dual modality system or dual probe. That's yet to be seen. Yeah, that's a good question. Thanks.
Hello, thank you, sir. That was a very insightful talk of yours. You
Thank
briefly,
you.
I am Divya Khatri, currently pursuing PhD in ocular drug delivery. So, sir, you briefly mentioned about ocular biosensors that track, that's related to my work also. So, I would like you to give a brief insight in that too, if that's possible for you now.
Yeah, so we're doing a whole lot. So we recently there's an agency called federal agency called ARPA-H. It's a Advanced Research and Health Agency. So our in the US, our primary funding agency for biomedical research is NIH, National Institute of Health. ARPA-H, this is another federal agency, and they started the programs I think 3-4 years back, and so this is very new. So they recently funded, they started a program around delivering drugs in eye and tracking it at the same time. So as part of that, what we were doing is we are developing a tiny sensor that can be placed inside the ear tear duct, and that will have a capability in a closed loop manner. It will detect biomarkers in the tears, and then it will inform a drug delivery module as a ring to release drug in the eye. So this is something that is very futuristic. It's looking. Right now, people are using continuous glucose monitor that 24 hours it measures someone's glucose. So imagine that that is connected to the insulin pump. So based on the glucose that goes up, then the insulin is being delivered, right? So this is all, they're connected to each other. So the same thing they're trying to do, and we were trying to really come up with strategy to do that same thing for ocular diseases. And more we're learning that the tears basically they're nothing but a byproduct of blood. So in some cases that the tears can be very valuable biological fluid. So it can provide us a huge amount of information that we were not aware of, including some of the neurological diseases, for example, Parkinson's disease and Alzheimer and so on. So this is one of the approach that we are taking right now. There are other ways to do drug delivery and I'm sure you know that because you're doing your PhD in that. There are other ways for ocular drug delivery, and for example, VEGF and VEGF targeted drugs, and those are all they really don't work in systemic applications, but they really work in for ocular diseases. So yeah, there are so many of these applications, and as a matter of fact, I think ocular drug delivery is one of the um, I would say one of the smartest way to do nanomedicine, because the clinical pathway and the FDA regulatory pathway is much different than the systemic drug delivery pathway.
Thank you, sir.
Sure.
Good morning, sir. I'm Shivani.
Hello.
I am currently pursuing PhD in nanomedicine. I'm interested in electrochemical sensors. So, as you have mentioned in one of your slides, that borofin has increased the LFIA sensitivity. So, I'm currently looking for a nano composite with one component is gold, and I'm looking for a biocompatible substance which can be used with gold, and it won't be hindering the effect or sensitivity of gold and conductance of gold. So, do you think borofin can be used without hindering the gold nanoparticles conductivity?
Yeah, I mean, yeah. So, Well, yeah, but the reason the question is why do you want to use borofin? So, if you're talking about conductivity, that means you're really talking about electrochemical, correct? So, if you're talking about electrochemical, the question is, why do you want to use borofin? I mean, you already have gold in there, right? So you can use gold. I'm not quite sure why you are interested in using borofin. I'm not quite clear. The reason why we use borofin for a lateral flow was that for lateral flow strips, the nitrocellulose, the way it works, that you cannot really put the antibodies properly. So the orientation of the antibody is not really proper always, so that when the target comes in, it binds in a proper fashion. So the spatial location of antibody is very important for lateral flow application. For electrochemical, I think it's a slightly different scenario. So I'm not quite sure why do you ask this question? Can you elaborate a little bit more?
Actually, I'm looking for a substance which can increase the biocompatibility of our gold nanoparticles and decrease its accumulation in our body for long-term usage.
Oh, okay. So you're talking about maybe a wearable sensor and those kind of things. So yeah, that's a good question. So there are multiple different ways we can do that, and borofin can be used because this is biodegradable completely. Now, but if you're really looking at something, say for example, wearable sensing and continuous sensing, that's not easy because you have to encounter the biofouling issues. You have to encounter the signal drift and lots of other things that you have to use. So biocompatibility is just not one of the things, right? So borofin can be used as a biocompatible material, but can borofin be used as a material that helps your cause in terms of the biofouling or the signal drift? Those are all yet to be seen, so we're not quite sure. I mean, we're also interested in exploring this material for wearable sensing, but we're not there yet.
Thank you, sir.
Okay.
So, good morning from us, and good evening to you.
Yes.
Very much for this late night talk that you have given, and this is really inspiring for all the students and raise some good questions. I'm Dr. Seema Bhatnagar. I have connected you through.
Yes, absolutely. And I responded back to you. Well, sorry for apologies for my delay, but I've been traveling extensively. Yeah.
So we know that you were connected with Dr. Tinku Basu for the research work, and as director of this institute, which I have recently joined past 6 months only. Earlier, I was with biotechnology. So we will continue these discussions. And we've got some new students, and you've raised some new questions in their mind. So we will definitely be in touch with you for.
Absolutely, happy to help, and we'll continue the discussion, and this is absolutely not a problem.
Very happy to have you today here at this workshop and also inspire the young minds. So I'll hand over to Dr. Rachna and she will take forward the proceedings.
Thank you so much. Yeah.
Thank you, Dr. Pan, for joining us today. Thank you once again, and we look forward to the future collaborations with you, as Dr. Seema mentioned. We'll stay in touch with you. Our students will keep contacting, and we'll look for your
Sure.
insights. Thank you so much for joining us today.
Thank you, Rachna. Thank you. Bye-bye.
So now we move on to the second talk of today's morning session by Dr. Mariam Rehman. She is from the professor in the Department of Neurosurgery, University of Florida. Due to some last-minute exigency, she couldn't join us in the session, but we have a recorded lecture from her. I'll briefly introduce Dr. Rehman is a professor in the Department of Neurosurgery, University of Florida. She received a bachelor's degree in chemistry from Johns Hopkins University and completed her neurosurgery residency from University of Florida. She completed her 2-year neuro-oncology fellowship under the direction of the world-renowned scientist Bernard Reynolds, studying tumor, stem cells, and their role in developing novel therapy for the brain tumors. Dr. Rehman Laboratory focuses on identifying novel immunotherapy approaches to overcome resistance in patients with the brain cancer. Current projects include developing vaccines for the treatment of brain tumors and combining immunotherapy with MRI-guided laser ablation. Her work has been supported by several grants from NIH and private foundations. Her translational research is actively being tested in human clinical trials, and she has published more than 100 peer-reviewed articles, multiple book chapters, and has been invited for several talks and recognitions. She was the first female president of the Florida Neurological Society and also the executive member of the Congress of Neurological Surgeons and also the executive member committee of the Women in Neurosurgery. So we welcome her and of course, feeling her absence. And I would now request to play her recorded lecture.
शेयर करना है ना एक सेकंड नोट करो
You guys are the warriors, and both Melanie and Clark, I really enjoyed your talks, amazing. So I really thought this was going to be kind of a counterpoint to Dr. Abdullah talking about how tumors are, so you're going to bear with me, but I'm going to go through some data here. My real disclosure is a lot of the work that I do is only possible because of all of the people that I work with, and I'll present some of the work that we're doing in the lab. Most people in the room know this, but brain mets are 10 times more common than primary malignant brain tumors, and so a lot of our tumor sessions are focused on gliomas, but brain metastasis in terms of cancer burden is a much bigger problem worldwide. About 200,000 people in the United States are diagnosed with a brain met each year, and this number is growing, as Clark said. The prognosis is still amazingly based on factors that I shouldn't say we, but people who came before me identified years ago, which is this RPA classification used for our TOG studies, which really identified that KPS, or functional status, is critical for prognosis. So anyone who has a good KPS, greater than 70, is relatively young, controlled systemic disease is class 1, class 3 is a KPS less than 70, and then class 2 is everyone else. And these are the survivals, even with the small molecule inhibitors and immune checkpoint inhibitors and all the stuff we have on the market. If you have a brain metastasis and you get classified as RPA class 3, you have about 3 months to live. And so it's still relatively poor. The rationale for surgery was established by Roy Patchell and others in the early 90s. So in a seminal paper that was published in 1990 in New England Journal, they took 48 patients with a single brain metastasis. This was a randomized controlled trial, and they were randomized to biopsy followed by radiation versus surgical resection followed by radiation. And this was whole brain RT in 36 gray. And basically found that patients who had a biopsy alone had a 53% recurrence rate compared to those who had a surgical resection, which was only 20%. And the overall survival was quite improved in the patients who underwent a surgical resection of their brain metastasis. And this was statistically significant. Importantly, these were the data of patients who actually died a neurologic death as opposed to death from their systemic disease. And then the survival advantage of the patients who underwent a surgical resection was even more profound in the surgical group, having greater than 62-week survival of a neurologic death versus the radiation alone group that had a median survival of 26 weeks only. In a subsequent study just 4 years later, they, same thing, took 63 patients, randomized controlled trial, single brain met surgery plus whole brain in 40 gray versus whole brain alone. Again, found median overall survival of 10 months when you added surgery to radiation, when it was only 6 months with radiation alone. And the greatest benefit was when their systemic disease was inactive or controlled, which really kind of puts them more in that RPA class 1 category. And so if you look at kind of this time period, these were kind of the major randomized controlled trials of comparing surgery plus whole brain versus whole brain alone, showing a clear advantage in patients who underwent a surgical resection. This was a very select group of people, right? Patients with a single brain metastasis. A lot of the patients we see do not fall into that group, but these are the data supporting resection. Obviously, a huge role for radiation, and I'm going to briefly go. This is the one place where we have class 1 evidence, randomized control trials. And so surgery plus whole brain is superior to whole brain. We just showed that. SRS plus whole brain is superior to whole brain, and Doug Conciocco had shown that for local control rates. Surgery, and again, these were all randomized control studies. Surgery plus whole brain versus SRS for whole brain were relatively equivalent when you're talking about brain metastases that are 3 centimeters or smaller. If you have large mets, then surgery is usually the only option. And then SRS alone, and you take out the whole brain, has better neurocognitive outcomes compared to SRS plus whole brain, with the caveat being that your brain disease is not as well controlled. You're more likely to develop a MET moving forward if you forego whole brain. But the idea is that what most institutions I think would do, we definitely do this at UF, is that SRS followed by more SRS is a reasonable option to achieve local control, even though whole brain will achieve better local control, but you're buying that by destroying kind of neurocognitive outcomes within a year. And so for oligomets, that tends to be our approach. But obviously for a tumor like this, big, a lot of edema, really the only reasonable option if somebody has a good KPS is surgical resection. Let's talk a little bit about the other options here. So we talked about surgery, whole brain, SRS. I'm gonna go through these others here. So gamma tile, there is no data like we have for surgery and radiation. There's no prospective randomized studies. This is essentially, collagen with the cesium-131. It has a short half-life, about 9.7 days. It delivers 90% of the dose within a month. The studies are quite small. They're a prospective single-arm, low number of patients, basically demonstrating that it's feasible, it's safe, They report, durable local control. I'm not sure if we can really say that based on the number of patients that has been reported in the literature, but it's out there. Small molecule inhibitors are becoming important for patients with brain metastasis. Melanoma was the case study for this. So the BRAF inhibitors were found to have intracranial response rates of up to 58%, but it can be short-lived. This was dabrafenib and trametinib. And then the immune checkpoint inhibitors, especially if you use dual immune checkpoint inhibition for melanoma, you can have efficacy for asymptomatic smaller brain mets. 26% of patients had a complete response, 57% had some clinical benefit. So these can have a role in patients who have small asymptomatic little lesions that are being followed very closely. And this has also been shown in other types of cancer. Lung cancer, the TKIs or the tyrosine kinase inhibitors, there's randomized controlled data showing that these by themselves or in combination with pembrolizumab and platinum-based chemotherapies can offer CNS protection and improved intracranial response rates with improved overall survival, and similarly in ALK-positive disease. These have been shown in phase 3 trials that have all been published in Journal of Clinical Oncology or New England Journal of Medicine. So there is a role, and this is a big change for us in brain metastases because this definitely requires an interdisciplinary approach where you are co-managing these patients with medical oncology. It's not just run all by rad-onc and neurosurgery as it has been in the past. Dr. Chen spoke very nicely about LIT, obviously a surgical option for patients with brain metastases. It has direct cytotoxicity to the tumor cells and has an additional side effect of temporarily opening the blood-brain barrier, which you could potentially take advantage of. Indications, again, probably similar size to tumors that you're treating with radiosurgery. Once they get large, you run into two challenges with LIT. One is that you don't get great coverage. Dr. Chen spoke about how if you don't really cover the entire enhancing portion of the disease, you don't have as good an outcome, and you get a lot of edema post-LIT because of the nature of the thermal ablation, and so managing that and making sure you don't have too much toxicity. It does allow for tissue diagnosis, which is nice. So in patients where you're questioning what it is this tumor recurrence versus radiation necrosis, you're getting that biopsy at the time of LIT. There can be sampling error with that, but still can be helpful to guide treatment. And LIT treats both recurrent tumor and radiation necrosis. So super helpful if someone's had radiosurgery and now they have a growing enhancing lesion in their brain. LIT is somewhat ideal for that. I would personally say that for us, primary treatment for metastasis smaller than 3 centimeters is still radiosurgery. There's class 1 evidence to support radiosurgery for the treatment of small brain metastases. And really for my own practice, I really use LIT at the time of recurrence after radiosurgery. And just like Dr. Chen said, there is a significant number of patients you will see where there's a question if it's radiation necrosis or tumor progression, and then LIT is kind of ideal in that scenario. We had questions in my lab about how we could potentially use LIT in combination with other therapies, and so we established a LIT system in the lab with the mice. And Anjali, who's the senior scientist in my lab, started doing experiments implanting tumors. She started with B16F10 ova, which is a melanoma model. It has limitations, but in this particular scenario, it was useful to us. And then treated them with LIT, and then subsequently obtained tumor tissue and the draining lymph nodes to analyze what happens from an immunologic standpoint. She found significant upregulation of our CD11B positive microglia/macrophage population within 2 days after LIT, and then 10 days post-LIT had a big infiltration of T cells, and specifically effector memory and central memory T cells. And so that's something that we're now working with to see if that's something we could leverage with immunotherapy. The other kind of portion of this, which has turned into a really interesting and productive collaboration, is Tim Spicer is a professor at UF Scripps who does high throughput screening. And so when we take the tumors out, from the operating room, we send them to him. He is able to load them without turning them into organoids into these 1536, very low attachment well plates, and generate high throughput screening results on 194 drugs that are all FDA approved in the NCI database, and then can tell us, based on CC50, which drugs are most efficacious against that particular tumor. That's fascinating, except we can't use any of these drugs because they don't cross the blood-brain barrier, right? Well, we thought, well, maybe we can take this high-throughput screening platform, combine it with LIT, since it does temporarily open the blood-brain barrier, and get some efficacy of these drugs. So to test to see if this was even feasible, Grace Hayes, who's one of our really talented medical students, first wanted to look in a murine glioma model if she could show that LIT increases the brain penetrance of a drug that we know should get into the brain, which is temozolomide. So she took mice, implanted tumors, treat them with LIT, gave them temozolomide, and then sacrificed them. And then we did LCMS analysis of their brains and did find significant increases of temozolomide at a very short time period post-LIT, within an hour, but did find that improved compared to TMZ alone. We then took a drug that has very poor brain penetrance, but is very easy to measure using, this time we used ICP-MS, and that was carboplatin. So, same thing, LIT, carbo, measure drug levels in the brain, found significantly enhanced drug levels within the brain when you gave it post-LIT, and then did a survival study and found that the combination of the drug plus LIT, especially if the drug was delivered neoadjuvant, like right before LIT, was significantly enhanced in this aggressive glioma model that we had. And so we have just now established our PDX, and so this is a human melanoma brain metastases that I sent to Tim. He did high throughput screening. We've now established this in our NSG mice as tumors. You can see this like fungating melanoma tumor coming from the brain, and now we're currently testing it against the drugs that were found in the high throughput screen. I will say these drugs are incredibly toxic. Like, this car fills a bit, like dealing with these drugs in the lab, we're like, whoa, this is bad. So if we were going to do this in humans, we would definitely need the help of medical oncology. So in summary, brain mets are associated with limited survival. Mainstay of treatment should be surgical resection, radiation for larger lesions, smaller lesions. There may be a role for systemic treatment, especially for melanoma, non-small cell lung cancer. LIT is effective for both radiation necrosis and recurrent brain metastases, and there is a potential for LIT with systemic treatment, which we're currently studying in the lab. Thank you.
Thank you, Dr. Rahman, for these insights about the brain metastasis. So the participants can get in touch with her for their questions over email. Thank you very much.
After the series of inspiring and insightful talks by eminent scientists and experts, we would like to thank Dr. Rachna Mehta for her time and consideration to chair this session. May I now request Professor Dr. Seema Bhatnagar, Professor Ashish Mani, Dr. Chansi, and Dr. Deepika to come forward and felicitate our session chair, Dr. Rachna Mehta, with the sapling. So may I now request all the participants to please move forward for the next session, that is the workshop session. Before this.
So we'll continue the workshop session after 15 minutes. We are meeting in lab J2304. All the participants, please be seated here only. We will take the attendance first, then we will move to the lab.
I'll just interrupt. I'll have to interrupt. I think we have to start the session because it's live. Session will be live soon in 2 minutes.
With a very warm afternoon to everyone present here and all our enthusiastic participants. We would now start with the next session. Before that, I would like to invite, it is my proud privilege and honor to welcome our esteemed speakers for this session, Professor Dr. Zeenat Iqbal and Dr. Himanshu Baveja. As we now proceed to the next exciting session of our speaker talks, promising another engaging exchange of knowledge and innovative scientific perspectives. I now request Professor Dr. I would like to request all the invited speakers and session chairs to please come forward and take your seats. Professor Dr. Zeenat Iqbal ma'am, Dr. Himanshu Baveja sir, our session chair, Professor Tanveer Naved sir, and Professor Dr. Ashish Mani sir. So please come forward. I would also now request Professor Dr. Seema Bhatnagar and Professor Dr. Nidhi Chaudhary to come forward and welcome our first speaker, Professor Dr. Zeenat Iqbal ma'am, with the sapling. I would thank you, everyone. I would now request Professor Dr. Ashish Mani and Professor Dr. Madhumita Ghosh to please come forward and welcome our next speaker, Dr. Himanshu Baveja. Now we would like to proceed to the next session, and this session will be chaired by Professor Dr. Tanveer Naved, Amity Institute of Pharmacy, AUP, along with Professor Dr. Ashish Mani, co-convener of the conference, Amity Institute of Innovation and Design Center, AUP. May I please invite Professor Tanveer Naved to please come to the dais and take the proceeding.
A very good afternoon to one and all. At the outset, I'd like to thank Dr. Seema Bhatnagar and her team for organizing this 3-day workshop, very relevant to the pharmacy faculty and students also. And my personal thanks to giving me the opportunity for this session. This session, we will have 2 invited talk by 2 very distinguished persons who are already present in the hall. Firstly, I take great proud and privilege to introduce the first speaker of the day, Professor Dr. Zeenat Iqbal ma'am. Professor Zeenat Iqbal is a professor of School of Pharmaceutical Education and Research at Jamia Hamdard. She has over 3-decade experience of teaching and research. Dr. Zeenat has published over 200 publications in reputed journal with H-index more than 50 and citation nearly 10,000. Her area of research has been mainly in the domain of cancer, periodontitis, novel drug delivery system. She has been administrative post for many coveted position at Jamia Hamdard and other professional bodies also. And One thing above all, I can say that I am one of the proud student of Zeenat ma'am. She taught me in the BPharm. So it's my personal honor and privilege to invite ma'am for giving the talk on nano intervention for women reproductive cancer, leveraging AI for improved outcome, a very relevant topic. May I request ma'am to please come for the talk.
नहीं नहीं ये वाला दूसरा दूसरा ये वाला ना बिस्मिल्लाहिर्रहमानिर्रहीम so much. Bismillahirrahmanirrahim. Shubh Aprahan. Salam. And a very, very good afternoon. So all the blessings of Almighty on us and on a very blessed day. I trust my voice is reaching to all ends. Thank you so much. So I am a bit overwhelmed, and I think nothing better than this can happen to a teacher. When one of your student is a dean, the other students are heading departments and doing wonders. So I think this is something very humbling also, and it is one of the, and as I think we are talking here, this is the only achievement of the teachers. We are actually poor, but we are very rich by these standards. So thank you so much, Dr. Seema. I have no words to acknowledge your invitation and the way you have invited me. I have to mention Deepika for being a conduit in this. And I'm overwhelmed, and I will start with an Urdu couplet, and which is maybe dedicated to the Hamdardians who are making a lot of light here and there. So it goes like this. So चाहे अजम हो चाहे अरब हो चर्चा है तेरे नाम का हर तरफ फैली हुई है रोशनी हमदर्द की. So that is the point. And I think MET and we share a lot of ethos. So whenever you talk of METians, I think the same feelings would be there. So it's dedicated to both METians and Hamdardians. Wherever you go, you just be blessed and let the light of everything comes on us. Your light will definitely keep a shadow on us. So the topic which I am going to intervene, or rather going to deliberate upon, is something very close to my heart. As a researcher, it's a long 2-decade journey where we had a lot of research work, and I'm very happy to share that Jamia Hamdard has been a pioneer when it comes. Oh, Farah is also here. Good. So Jamia Hamdard has come as a pioneer in the area of nanoformulation. It was around 2006-2007 that the government of India launched Nano Mission. And one of the projects which was given to us at that point of time led to a lot of work in nano. And since then, we are continuing our work there in this particular area. So I think close to a decade and a half, we have dedicated to the nano formulations and their design and evaluation and all. So over the time, as Dr. Tanveer put it while he was introducing me, so we have taken up some verticals. So the verticals which are of interest to us, and depending on whatever PhD scholars are interested in. So we work in 3 basic verticals. The first one is your dermal delivery, which is again appending the various nano formulations. And I'm being a little elaborative here for the interest of the PhD scholars and maybe future collaborations if at all we can do. So we are working on the area of dermal formulations, and then we are working on a very niche area of periodontal infections. So and I did my PhD on that. That's very close to my heart. So we have developed a lot of intra-pocket delivery where we have been able to disrupt the biofilms constructed by the microorganisms by using the domain of nanoformulations. And the third area, which primarily some of it I'll be sharing here, is about the nanoformulation or pharmaceutical interventions, which are covering the reproductive and particularly women health. And the area by default would include cancers, which are like breast cancers or your cervix cancer or ovarian cancer and so on and so forth. Currently, 2-3 students are working in this area. So with the permission of the chair, I would try. May I start, ma'am? Okay, thank you so much. So I think I'll skip this. So I think needless to mention, we have a doctor in the house. So when we talk of cancer, so we know that it's an uncontrolled growth and it is being associated with certain attributes which are not present in a normal cell. So something goes wrong in the body and it gets and develops into something which we now refer to as cancer. So the other word which we use is tumor, and then we have a lot of classifications associated with it. The global standards primarily, I usually, when we talk in the research groups, I say that cancer is a great equalizer. It doesn't recognize the celebrity. It will not leave a slum dweller. It will be, unfortunately, each one of us are as susceptible as the other. So these are some of the slides, and using the celebrity faces sometimes helps us to understand that this disease has surpassed the societal strata. So we have to be very cautious whether we are from the highest echelons of the society or whether we belong to a maybe suburban or a rural population. It is absolutely important for all of us to be aware of this domain. So some of the celebrities, I think most of us are recognizing all of them, and they have been fighting cancer. We lost some of them like Jade Goody, but others have come out cancer-free. And one of the main ways and reasons why they could be cancer-free was the contribution of the pharmaceutical industries or us who have been able to develop such good chemotherapeutic agents. So I think this is again a very important phase. She primarily she went beyond that. She was not having the cancer, but she had a suspicion because of the reason which is known to all of us, that maybe because of somebody in the family or the maternal side was already exhibiting those genes. So she underwent something which is referred as gene profiling, and it was found out that she has high susceptibility towards the breast cancer. The BRCG1 one gene is present there, and maybe by the age of 40-45, she might go in for the cancer. So the use of AI is like long back in US, and the gene profiling and the concepts which are newer now were primarily the basic driver, and they helped her to take a decision which is a very radical decision of getting a double mastectomy. But then we are unfortunate this side of the world that these types of interventions are still far away from our reach, and we cannot go in for a futuristic understanding of this disease. Nonetheless, there have been a lot of buzz in the pharmaceutical industry, and there have been a lot of construction of the chemotherapeutic agent. So that's where we can take a pride. We are already the pharmacy of the world. We are already in volumes. We are the fourth number in the global standards. So that is the area which the government of India is also supporting. And a lot of medicines and chemotherapeutic agents are primarily being just, I would say, formulated, and research is going on. So that's the statement which I already made, that beyond the social strata, we have got a lot of sufferers across the globe, across the world. So this is something which primarily, and unfortunately, there has been a very huge rise in the presence of the cancers. I usually, when we discuss, I usually have that feeling, and I do not hesitate to put it up here, is that earlier maybe the awareness about the detection was not there. So the cases must have been then also. But now, because we have this machinery for detection and the government policies are there, so the unraveling of the data has come up with a big surge. And I think Dr. Himanshu, being a clinical researcher, would agree with me that because of the usage of the early diagnosis and detection kits, and then in advent of the artificial intelligence, their tools and so forth, have led to a very good situation that we are able to unravel the cancer. But if I may just put it very simply, that the cancers which are of reproductive in the females can primarily be based on the anatomy. So it could be an ovarian cancer or a cervix cancer or uterine cancer or a breast cancer, to say the least. The cancers primarily, just in addition to what I have just mentioned, that we are, I think all of you must be aware that the government of India has actually put a lot of efforts on the cervix cancer vaccines, and which has to be given at girls after this menstruation starts and up to the age of 21. So the major reason was that this HPV is becoming one of the major key drivers of the cervix cancer. So if we have got a vaccine against it, so maybe the numbers would reduce, and that is the intention of this whole program. So as we move further, we are just going through the incidences. I think we can skip this slide, but the situation is pretty bad. The number of incidences in Indian scenario are as bad as in global scenario. We are not anyway being saved from it. So you can just appreciate from this slide that primarily this sort of detection of the breast cancers or the ovarian cancer is something which has come by a storm onto the earth. And a lot of people, the data is up to 2025, and the data is still surging up with a lot of cases which are emerging. The basic thing as pharmaceutical scientists and primarily from the medical field, we as students as well. So one of the things which should be very clear to us is that what makes us susceptible to a particular diseases. So we usually have a very technical words that what are the risk factors. So this slide has been like put deliberately just to give us a little hint that sometimes the things which primarily are ignored, risk factors could be as simple as the breast puckering, which is being avoided. Risk factors could be as simple as a leg pain and swelling, and ultimately that becomes a big sign that the person could suffer from cervix cancer. Something as common as an abdominal pain and bloating could be a sign of as bad as an ovarian cancer. And sometimes as simple things like being a simple fatigue and having that thakan, we are very commonly taking it and ignoring it, that a lot of fatigue, we work a lot, and that becomes a sign for the uterine cancer. So we are all, I think, a lot of women in the house who are of this reproductive age, and maybe families also have a lot of from the fair gender. So it is very important for us as student of pharmacy that we should be aware of this risk factors and we should not ignore them. So that becomes a domain for both the doctors as well as the pharmaceutical researchers, that we understand what brings the disease in. Then now coming to the little bit of technical part, and I'll be very interested to interact with the PhD scholars even beyond the presentation. So in a nutshell, if I may say so, whatever this thing, breast cancer or uterine cancer, ovarian cancer, if you just bring it to one logical say or a logical term, they are more or less the solid tumors of different varieties, different types, and different challenges. So the solid tumors and the liquid tumors, as the name suggests, are different. And ones which are impacting upon the reproductive cancers are in the category of the solid tumors. They have got their own ways and mechanisms by which they progress. And as a pharmaceutical researcher in the area, and maybe the budding researchers here, so we need to understand the biology of the disease or the pathophysiology of the disease. If we understand the underpinnings of the disease, we are able to give a solution to it. If I know that something is going wrong, there is a problem of high level of oxidation, then I know that antioxidant can be a supplementary thing. Similarly, if something is going wrong in the disease in a particular mechanism, so I can choose my drug. I can choose my drug based on those particular symptoms, not the level of symptoms which I'm seeing. I'm talking about the molecular mechanism which are involved in that disease. So these are the current, needless to mention that before the nanoformulation and even in today's form, because of the limitations of the scaling up of the nanoformulations, we still heavily rely on the conventional treatment modalities. The one which is the most feared is the radical surgery, which may have different types of aspects depending on what the disease status or disease condition is. And the doctor primarily, they are the oncosurgeons or oncogynae surgeons nowadays, super specialties available, who would decide what action should be taken. Chemotherapeutics is again a mainstay, and the categories which are of relevance to us are the cytotoxic drugs primarily, which we deal with. Then we have the biological response modifiers or disease modifiers as of now. Differentiating drugs are there, and anti-angiogenesis drug is there. The take-home message from the slide is that depending upon the condition and depending upon the type of treatment which is demanded by the patient, we will have to have a mixed bag of these therapies. I think Dr. Himanshu will agree with me that the hospitals usually have an onco board which actually decides what type of personalized treatment has to be given. So all this armamentarium which is available to them is again very deeply scrutinized and based on what is the requirement of that patient. We cannot have a generic thing for all the patients right away. Then miscellaneous include, I think, radiotherapy is again the targeted therapy, the immunotherapy are the new advance in this particular area. Just a snapshot of the various drugs which are very particularly used for the reproductive cancers. Now the question comes that I will proceed from a conventional drug delivery to a sustained release or a targeted delivery if I find some fault with this. I cannot unnecessarily jump from a normal state and then go to the novel drug delivery. So there are certain very key challenges which are there with the conventional therapy. I would not primarily delve deep into it, but we know that because of a lot of issues, there is the limitations which are associated, which can be low bioavailability, which can be a problem of reaching to that deep organ, which can be a problem, both pharmaceutical as well as delivery, and so on and so forth. So the chemotherapeutic agents, needless to mention, come with a package of a lot of side effects. So we have the gonadotoxicity, and in young female patients, one of the key areas which is of concern to us is that the fertility gets compromised. So there is a very dedicated field like oncofertility area, which the doctors try to preserve their fertility. So we take a hope from this side, or we rather take a driving force from here, that can we do something as researchers to just maybe reduce these side effects and maybe make the survival of that patient much better. What can be done? So we delve into this particular question, and that is primarily which brings us to our area. The slide and the central part is very close to my heart, and I'm sure it must be close to everybody's heart, where we talk about the 12-year journey, putting up billions of dollars of the money into getting just one drug. And that one drug primarily is, it doesn't come like this. It doesn't descend from the heaven. It comes from the preclinical studies and the clinical trials. And I think Dr. Himanshu, I'll call on him again, that he knows that how much of energy and money gets pumped into it. And that one drug comes into my hand. So that drug primarily I will hold very closer. I will launch it in a different manner. And till I am able to use it, I will try to use that drug for 100 years. Once in the form of a tablet, then in the form of an injection, maybe in the form of nano formulation, and so on and so forth. So this whole concept of a new drug discovery, one thing which is, Dr. Ashish is here, I think he'll agree with me, that this whole journey of 12 years is becoming a little easy on us by virtue of adapting something which we refer to as artificial intelligence. So we are able to leverage the artificial intelligence to cut down the time and maybe bring the product, not one at the end of it, maybe 5, 10, 15, which are having much, much, much more success. So that's again a driving force and motivation for all of us. And here we go. The journey which I was just saying, just a little bit of recapitulation. As a student of pharmacy, we had been always talked about and we are being like acquainted to something which we refer to as the conventional dosage forms. We do not rely on this, and we move to the next big thing. So once we are having the solid dosage forms, be it the capsules and the tablets, the next thing what I can do, there are a lot of limitations like patient non-compliance and so on and so forth. We all know it very well. Then we move to the next big thing, and that was something referred to as your microparticle drug delivery. We did not rest in peace. We moved further, and then we moved in an arena which we refer to as the nano formulations, or the concept or the birthing of the nanomedicine came. And we are not resting in peace. I think Dr. Seema would agree with me that we cannot rest in peace. We'll try to do something that, okay, can we repurpose an old drug and make it into a nano formulation and then use it, which will be having less side effects and so on and so forth. We are continuously going on this journey. And one of the things which is again come to our rescue is that I need a ball. So one of the recent things which is happening is that we are having this concept of repurposing of the drugs. So the drugs which have been there, for example, I give a key example of metformin. We all know metformin to be an anti-diabetic drug, but it has shown a huge amount of potential for treatment of ovarian cancer. So these are the things which as researchers, we are capturing from what the research area is offering to us, and then using them with our expertise of maybe formulation, in this case, nano formulation, and then utilizing it for the best outcomes for the humanity. That's exactly, I think that's a continued mode of motivation for all of us who are in this hall. So as we move further, this is a slide which I would dedicate to all these gentlemen who are not from pharmacy. I always joke with my students that pharmacy is very easily accommodating things from other fields. And then we accommodate them so well as if they are our own. So this is a complete, nano as such is a complete physics-based thing. but we have accommodated it and we have birthed something new, which is called as a nanomedicine. And they are always in okay, this physics part has been used for the delivery system, and we are doing it very successfully. But I think the credit goes to all these gentlemen who have been guiding us through this. So here is something which I always believe: small is beautiful. And in case of the nano formulation, beautiful pharmaceutically, beautiful pharmacologically, beautiful when it comes to the patient compliance, and so on and so forth. The basic concept still remains that we have a simple system which is small, and we have a particles which are of dimensions of the nanoscale. You can just have an idea of the dimension, that how small that nano formulation can be, as small as 10 to 1000 nanometers. But we usually play between 150 to 200 nanometers to get the best outcomes. The structures can be in sub-nano size, and so on and so forth. So this primarily boils down to this very basic concept that we had a drug, because the drugs which were in the conventional therapy are naked or neat drugs. They enter into the blood flow or the bloodstream. The blood is not very happy to receive them. It will react in a manner which is known to us in pharmacology. So what we are doing is that we are simply clothing it up, or we are mantling it up, and we're creating a drug-loaded nanoparticle, and which has been coming with all these types of advantages. So we are able to reduce the toxicity, we are able to enhance the solubility, whatever we want, and increase the drug targetability, and so on and so forth. A little bit of timeline that we had talked about: the cancer nanomedicine as old as 1964, still not very old. And since 2015 and beyond this decade, we are continuously working on the advancements of these nanoformulations. And primarily, I'm talking only at this point of time. Of course, I would not be saying that it is only limited to the cancer, but majorly cancer is the area where the nanoformulations are very, very well received. Then these are some of the areas, and particularly for the attention of the students in the house, that when we talk of nanoparticles, is a huge nano armamentarium which is present there. It is our research acumen which helps us to decide that what type of domain do we enter. Primarily, if we, just for the ease of understanding, we can have something like the polymer-based or the lipid-based and the non-polymeric and the nanocrystalline. So primarily, each one of them a huge amount of difference. They're all available. The only key driver has to be that whether they are able to complement what we want to do. It should not be that I need a liposome and I'm preparing a polymeric nanoparticles. I am needing something like, for example, nanofiber. A nanofiber cannot be given orally, and I'm making nanofiber for oral delivery. I usually go in for DCs to the other universities. So one problem which I usually cater to is that the students, because they are enamored by nanoformulation, they select any nanoformulation, whether it is suitable or not. I have a lot of arguments with my students. They will say that I want to make nano. And my question is that why? Because at some point of time, again, I think the medical person in the house will agree with us that there are situations where the cancer does not need that the drug enters into that cell. There could be simple angiogenesis which is happening, and we cut that angiogenesis. It will result in the shrinking of the tumor. Why do I need to have a small particle? This can be done by a submicronic or a microparticle as well. So one thing has to be borne in mind, that we should be able to assess that what is the requirement of the disease and the pathology. So I usually stress upon this. I'll again reiterate it. Understanding the pathophysiology and knowing the pharmacology is the key to being a good formulation scientist. If I am able to have the impetus from these two areas, I am going to definitely deliver a drug delivery system which is going to do wonders in the human system. We move forward. I think this is a showcasing of the various types of formulations. These are some of the slides which are dedicated, the next few slides, just to give an idea that mostly they all have coming under the umbrella of nanosystems. But each one of them, as you see, the internal part of it are designed differently, and that design will be then used based on what type of drug I am taking, whether it is a hydrophilic drug or a hydrophobic drug, whether I want a low payload or I want a high payload, whether I want an anchorage with a antigen or do I need something more. So all these designing are there for us to understand, and based on these designing, I will select whether I go in for a dendrimer where I need a huge payload, or I restrict myself to a polymeric nanoparticle, which is a small matrix which will take my drug without getting, I would say, attacked by the things in the blood, and ultimately reach to my tumor and go deep down into it. So, and the sphere shape, I would not go in for the cube shape. I'm going in with the sphere shape because that is the majority, the size to the volume ratio, and that is the one which is most stable, and that would enter into my tumor much more easily. So I would have to take into consideration everything which is being told to me technically as well as pharmaceutically. Again, the same area, just a little bit of extension of what I was talking. So these are some of the 3 main formulations which are very suitable for, let me say, if I may use the word, that they're more suitable for the hydrophobic drug. Again, the chemists in the house, if primarily I'm going to make a statement with a lot of responsibility, that as we are making new drugs, most of them are hydrophobic in nature. And by virtue of their chemistry, they are hydrophobic in nature. I have no choice. They come to me like this as a pharmaceutical scientist. So I have to find a way that how I can entrap them. So I know that a lipid-based particle will be an easy task in this, and I make nothing else but the liposomes or the solid lipid nanoparticles. I think Saurav worked on the solid lipid nanoparticles. So, and similarly, I will choose for the liposomes. I may not choose. Liposomes is very easy for the topical preparations. Most of the cosmeceuticals which are now available are liposome based, and some of them are also used for the skin cancer. Nano emulsions, I think the safest game where we can incorporate both the hydrophilic drug as well as a hydrophobic drug, and how they're constituted, easy to make, very stable, and so on and so forth. So here we have another area, another formulation, that is neosomes and the artificial exosomes. As the word zome suggests, most of them are spherical. So there will be preparations or formulations which are having the drug which is hydrophobic in nature. I have no choices but to heavily rely on the choice of the surfactants. So neosomes is a system, I would say it is a descendant of liposome only, but the amount of surfactant which is used in a neosome is helping me to increase the payload of that formulation, and maybe it is helping me to stabilize that much better. So I go for neosomes and not liposomes if my payload has to be high. Similarly, artificial exosomes, I can take it from the blood sample as well. These are usually the excretory bodies of the blood. They're present freely in the blood. So I can actually extract them. They're also available in the milk, and both of them have got a lipidic structure, and I can very happily incorporate the drug which are of hydrophobic in nature. Then non-polymeric particles, this usually I do not enter in as a researcher, because this is my personal opinion, that gold and silver nanoparticles are primarily unnecessary, adding burden to the human system. They are non-biodegradable, and by virtue of biomagnification, they may harm in the long run. But they have been very nicely used for the concept of theranostics. They are very highly used for the concept of simply taking the nanoparticles which are made up of gold, and they can be targeted to a tumor, and then by virtue of the photodynamic therapy or the other tools used, they can be used for a very early diagnosis. And I can combine the therapy with that. That is why they are very suitable for something which we refer to as theranostics now. I hope my speed is okay. I speak very fast. It's working for all of you? Thank you. So then the quantum dots and the nanotubes. Nanotubes, we had started work in around 2008, but what we found out in our laboratory, that by virtue of coming from an inorganic source, it has a tendency to kill the normal cells as well. So we stopped working on that because our intentions were different. So and it was like a chance discovery that pure nanotubes were used, and we just had a comparative. So the cytotoxicity with the pure nanotubes was also 15%. So that was not a very good sign for us. So we are not working on this area anymore. So these are nano tools which are available. Like a very true artisan, will have to keep on sharpening them and keep on adding to it what can be done. So decoration of the nanoparticles or surface modification is the next advancement there. Now, as I just mentioned that we had been working for 1.5 decades or so. So one of the key question which comes from the students, and we believe in a huge, at least our research group, we argue a lot. The teacher and the students, they'll argue a lot. So one of the things which the students had been continuously asking is that we have been doing wonders when it comes to research and preclinical data and all. Why do we not have enough number of formulations in the market? Why nanomedicines fail? I think it's a very generic question. It crosses everybody's head. Deepika has spent 4.5 years on it, that why it is not coming to the market is a big question. So this nanomedicine primarily fails. So we wrote one review article on it. This review article was of around 40-50 pages. And we tried to bring up that all the reasons and hurdles why this nano system is not favorably going there. So while we were digging for this article, one thing went very good for us is that we understood what are the hurdles, and we started designing our formulation based on those hurdles, so that this hurdle will be overcome if we design it that way. So this review article was a big watershed moment for our research group at that point of time. then these are the challenges again that it is. And one more thing which most of the students would be complaining is that when they do their research on a cell lines, and that's a 2D structure, the results are all thumbs up. Okay, wonderful, cytotoxicity has happened. We take it to the animal model. So results are little humbling down. No, it was 100% toxicity. Now it is showing a little tumor regression. And God forbid, if we directly take it to the clinical trials, we will have to actually come back. So there is a hurdle somewhere. So one of the key reasons which we should understand is that when we have got a cell line culture, we have got a 2D structure. And the formulation which we are putting deep down is in direct contact with that cancer cells. So definitely no hurdle of entering. It will go, it will do what it will do because it's a cytotoxic drug. But the moment we are talking about a tumor, it is something like tortuous. It is of a different shape. बिल्कुल अदरक की तरह होता है. You don't know from which angle it is moving out. And then primarily these types of tortuous nature leads to a very compromised lymphatic vasculature. It will lead to a lot of issues there. So the penetrability, it will have a high interstitial pressure inside it. It will have hypoxia. It will have so many acidic nature in that. And that becomes a huge hurdle for the real success of the formulation. So again, the key point from this slide and the previous discussion is that we need to understand these hurdles, and then maybe we target something which will just work on the microenvironment of these cancer cells. So here are the hurdles, just a little bit of listing of them. The primary hurdle is in the distribution. So the presence of the leaky vasculature as well as the lymphatic vasculature, which is compromised, and then presence of this interstitial fluid pressure does not allow the nanoparticle to actually reach where it should reach. Similarly, there is other barriers which are present there by the virtue of its size, the shape. Sometimes the shape is also not very easily being penetrated. So again, it is a key area that is not always to go in for that khandani shape of your spherical. We can always play with the shapes also. We can make them needle shape, and we can play with some cubic shape and so on and so forth. Every decision will depend upon the type of the. We move further, and primarily some of the other challenges could be the MPS system, which is always there ready to enamel. If the particle size becomes a little bigger than 200 to 250 nanometers, the phagocytic system will catch it. So we have to play between a particle size. The particle size, if it is small, around 8 to 10 nanometers, goes down elimination to the kidney root. So we have to manage it somehow that we are able to befool that phagocytic system, and that is being saved from the phagocytic system and reaches to the target, which is our tumor. Similarly, the circulation and stability becomes a big problem. So I think most of us who are from pharmaceutics background and pharmacy background will understand what I'm saying next, that when we are talking about a formulation which is hydrophobic in nature. Hydrophobicity attracts the phagocytes more because of the likeness and because of the recognition. So what we have been doing, we are able to do a concept of, or use a concept of pegylation. So that pegylation increases the hydrophilicity or decorate the surface in a manner that it will keep on circulating for a long duration of time without being taken up by the macrophages or the MPS system. And this concept is called as your stealthy system or the stealthy particles. So we can create pegylated or stealthy liposomes also, stealthy polymers also, and so on and so forth, polymeric particles as well. Then, as I just mentioned a bit back, the microenvironment barrier are huge because of the presence of the hypoxia, low oxygen. As we move from outside to inside, the environment will become more acidic and so on and so forth. So I read a very recent research paper where they have used normal antacids. So they are using type of drug, antacid. So when they are using this, the environment becomes a little. No, I'll come back, no issues. So it's like a teacher's job. Now you will not stay at one point. So this antacids like omeprazole and this rabeprazole have been used to reduce or increase the acidity. It is acidic, so bringing it to the normal thing. The moment it is around the same pH of the blood, so the transformation of the cell from a dirty cell to a normal cell becomes much, much higher. So just simple use of a drug like omeprazole along with the topicon, which is cytotoxic drug, can do wonders for the cancer. So I think most of the onco boards, I have been talking to some of the doctors, so they do take this into consideration. If need be, they also use antacids as a, I would say, supportive therapy sometimes. Depends on what type of cancer they are handling at that point of time. then one of the biggest major thing is that there will always be this extravasation. There's always the leaky vasculature, and there will not be a control that how it extravasate from one part to the other. And I will talk about it in the next slide, perhaps. We are having the passive and the active type of targeting. So the passive targeting, as the word suggests, I am not doing anything. I'm just putting up into the injectable route, and it goes by virtue of its being there in the bloodstream. But the moment it reaches to the tumor, because tumor is adhered to some tissues, of course, yes. So what exactly is happening, which you can appreciate here, that the vasculature or the blood vessels are not intact here. They are having the leaky vasculature. So the, I'm sorry. So these nanoparticles primarily will find its way into the tumor, and because of its tortuous nature and because of the lymphatic compromise, it will stay there, which is referred to as your enhanced penetration and retention effect. So otherwise, passive drug would not have been effective. But despite whatever the challenges they face, they do reach the tumor site. And in the tumor site, because of the pathophysiology of the tumor, the nanoparticles are received, but they are not let out. So they keep on rotating inside the milieu of the tumor. And that is the concept which is now recognized as an enhanced penetration retention. I may very quickly add here that the tumors, particularly in the women, and I have a research paper on the breast cancers, that all the tumors are not having the same amount of EPR effect. So they use the word hot tumors and cold tumors. So the hot tumors primarily have a nature where the vasculature is being sort of disrupted, and then they are more suitable for the EPR effect. So knowing through the artificial intelligence that what type of tumor that individual is suffering from, we can decide whether we can rely on the passive route or we have to move on to the other one, that is the active route. Needless to mention, when we talk of the active route here is that there will be some sort of liganting, there will be some sort of complementary liganting onto the particle surface, which will be then engaged with the receptors which are present. And tumors are normally known for overexpression of certain receptors. That could be folate receptors, or for that matter, transferrin receptors, depending on what tumor we are talking about. So if I know the pathophysiology again, I can do something complementary. So I take folic acid as a decorating entity, which will by default be attracting towards the folate receptors, and it will be taken up, and I call this as an active targeting. So active targeting definitely are more sort of responding. They are giving us better therapeutic outcomes as compared to the passive targeting. So we are trying to overcome all these challenges through the work which we have been doing in our laboratory. So the concept remains very simple, that we are now using the computer-aided drug designing, the molecular docking to find out what drug is being used. Then we are sort of unraveling the druggable sites. We go in for the pharmacology networking. And then finally, we go in for selection of that API from amongst the available APIs. We know that, okay, this is going to work for us. And then we know the disease well. So we unravel and the underpinning of that disease. We go deep down into the pathophysiology. And based on this complementary thing, we decide on what route of administration. So we have been talking about the first hepatic metabolism surpassing, for example, if you want to treat a disease which is not in the liver. But if at all we are having a liver cancer, then primarily we would be very happy that let it take it to the liver. So we don't need any targeting for that matter. In case of the cervix cancer, we know, okay, there are certain receptors, and we have been very recently, we are also thinking about using the concept of the HPV presence there. So maybe some antiviral drug in combination with the cytotoxic agent if it comes out neatly. So just to put, I am taking more time than what I am being allotted. So I will try to wrap it up. You get into your teacher mode and you forgot you are in a conference. I'm so sorry. So then I'm extremely sorry, Dr. Himanshu, I'm eating on your time, but I'll try to put it up quickly. So when we talk about the limitations of this, then there will be something which we refer to as a design concept. These are some of the key things that we have got a toolbox, and we have got a niche cancer interventions depending on what type of cancer we are heading. So normally, not only I will depend on my formulations, I will also have a complementary usage of HIFU and radio frequency and all, or ablation for that matter, or photodynamic therapy in order to get the better outcomes. We move further. And this is a slide which is giving us a lot of hope. It helps us that based on the artificial intelligence, I can stratify my biomarkers. And if I know the biomarkers that now you must be hearing a lot about triple negative breast cancer. ER negative, PR negative, and all the three HER negative. So we don't know how to do it. So if I am aware that what is required, if it is an EGFR positive, so I know that I will decorate it with something which is EGFR complementary. So I know that my drug will reach at that particular site. Then maybe I can go in for the tool like an immunotherapy. I can go in for the CAR-T therapy, and to get what therapy is suitable for what type of patient. The one thing which is helping us doctors and researchers is the use of the artificial intelligence, machine learning, deep learning, and so on and so forth. The time which we were spending on discussions and then having experiments has been reduced drastically, because we can have a huge prediction that this will be used in a better way, and this will be the therapeutic outcomes. So here are some of the other snapshots. I think I'll take a very quick exit from these slides. So this is just a slide which helps us to understand. So normally speaking, the T cells of our body are manufactured by Almighty God in a manner they would also attack cancer cells, because they also have the similar injury-like concept and inflammation-like concept. But something goes wrong while they are attacking, or they're not sufficient enough to that. So this concept of CAR therapy is hugely based on that we have a chimeric antigen receptor which is attached to the T cells, and this T cells, and then directly from the same patient, they are taken. Then this attachment happens in the in vitro conditions, and then again this is given back to the patients. And this CAR-T therapy is something which is changing life. And this is absolutely focused, because this T cells which are having this CAR will primarily only focus at the cancer cells, not hurting the healthy cells, and leading to the best positive outcomes. So similarly, the immune concept, very quickly, I'll just summarize here. Normally, the T cell has got something called as PD-1 receptor, or the protein which is expressed there. And this PDL1, that is your liganting for the apoptosis thing, is attached to the tumor cells. So normally when the T cells come happily to attack this cell, this intervention does not allow the T cell to be active there. So what we do now, that we get this anti-PDL1 entities, which we give ahead of in the concept of our therapy. And then this gets engaged. And now the T cell, which is being like redundant by virtue of this PDL engagement, becomes functional, and the same T cell therapy can destruct the tumor. So this is again thing that using from the patient's blood and doing the same thing right away. These are some of the customized therapy. I think we have dwelled enough in the beginning that what are the formulations which are available. And each one of them, I'll just very quickly summarize that some of them are suitable for cervix cancer, some of them are suitable for the uterine cancer. We have to just find out the pros and cons, which one is better. And based on the signaling, based on the overexpression and the underexpression of the receptors, we can just take change of these. These are some of the papers which primarily we have been using in our laboratory. My student Devina Verma, who was the first one to work on the breast cancer in my research group. Then these are some of the papers available on the net. We have got this concepts of magnetic nanoparticles. This is something which we used, that the nanoparticles are having the magnetite, and by virtue of an external magnetic field, they can just get collected into the solid tumor and deliver the drug right there. This is called as guided drug delivery. Some of the other papers supporting all the things which I have mentioned here. We still keep on asking questions. We are ready to fail. Currently, we are working on an area of nanofibers, which is a DST project, where we have got this machine, which is an electrospin, and we're trying to create a mesh or a mat, which we will be wanting to insert into the vaginal cavity and be attached to the cervix part where the cancer is proliferating. So these are the slides I have given a heads up while I was talking in this initial part, that this AI is used right from the drug discovery to the formulations. We are using different types of engagements. We are basically using it for diagnosis and detection to begin with, but needless to mention that we are also using it for guiding of our formulations as well. So using the AI tools is helping us to predict that how the formulation will work if it is being designed in this manner. So the concept of drug purposing, as I just mentioned, it will lead to, if I use an AI-driven thing, then the chances of success are much higher as we were doing in the conventional ways. These are some of the tools which we have been using, the machine learning and the drug delivery. As I just mentioned, that AI is now being very well used in basically of the nanorobots, and these nanorobots are not limited to only biomarker detection. They are now very nicely being used for the delivery of the drug. And the robots, as the name suggests, can be controlled from the outside. So you know where the swarm of the nanoparticles is to be sent. These are, again, I think we were talking a bit back about the plant derivatives, phytochemicals. We have started working on that as well. So where we are combining it with the cytotoxic drugs, which are synthetic. Applications in drug discovery, the things which I have already mentioned, and the target, I think, regulatory aspect of AI, maybe Dr. Himanshu will touch base. So these are some of the challenges, including the ethical, which we have to part. The infrastructure is there. These are the references which we have taken. These are the future directions. All the things which you have spoken, if you are able to do, we can get something like a magic particle. These are the nano tools we keep on honing, and this decoration of the nanoparticles using the viruses and all for delivery is something which is a key area. We have not started working in it, but this is one area which we look forward to. This is a little bit contribution of mine, whatever I did, the place from where I come. And I'm proud that it is a very empowering place for women. And these are my acknowledgements to all my students. This is the tree where I am at the bottom and all my students branched out. So, and this is my daughter, and women health really matter. Thank you so much for all the attention. And this is my a poem from Khalil Gibran, which always keeps me motivated. We have to stay fearless in whatever field we are. Thank you so much, and all apologies. I have actually taken a lot of time. Thank you so much. I'm open. I'm open to questions because I usually get into that teacher's mode, and then you want to explain more and more. And when you see, I was not looking at the faculty, I was looking at the students, and they seemed interested. So that's why I thought I'll explain a bit. I'm extremely sorry. I'm open to questions if they have any pointed question. I think Kavita and Saurabh.
If there are questions.
Should stand up. The reticuloendothelial barriers, if you want to cross, what should be the size?
Usually, ma'am, as a research paper say and as our own experience says that if we keep it very small, it is going to be a problem. And if we keep it very large, it will be a problem. So in order to befool that RES system, a size which I feel good is around 150 to 200 nanometers. But precisely around 130 to 150 does wonder.
But if we want to go through the vitreous side, intravitreal injection, there it would penetrate? Yeah, 100.
Organic nanoparticles, and usually it's a very good question. So when I have been working on polymeric nanoparticles, so there is always a question whether it was penetrate or not. But the concept of endocytosis works there. So the particle goes and attached to the outer cell membrane, and by virtue of its natural invagination, it will take it up. And then it merges with the endosomes and then the late endosomes, and finally it releases the drug. But precisely the size, because we have been working for long, so precisely the size which is helpful is around 100 to 150 nanometers. 100 dots. But unfortunately, I will say the negative side also, for polymers, it becomes very difficult to get a particle size of 100. That's a problem. So in that case, we are now contemplating that we may go in for hybridization, polymer plus lipids, in order to gain that side. And that comes with experimentation only, because paper says something else. And I'm afraid to say papers are not, we are not able to reproduce them. So that's a one big issue. That's it. Anyways,
so much.
Thank you so much. Okay, please.
The presentation was really wonderful.
Thank you so much, ma'am. It means a lot.
Yeah.
Thank you so
much. Well, I'm working on therapeutic enzymes and technical enzymes as well. So I need your guidance in how to take them forward. They have shown wonderful results in vitro.
Okay.
But now I am not into animal studies, but I need to take them forward so that
animal studies, if you're going in for pure enzymes, is not an issue. But if you intend to go in for formulation, enzymes and proteins per se are the biggest challenge on earth.
Yes.
For formulation scientists, the size will be a problem.
Yes.
Then enfolding of them will be a problem. But very recently, we have been, not our group, the other group in the Jamia Hamdard, and some of us are also working on that the protein delivery we are looking forward to, and liposomes are the best. But the stability becomes an issue. That's there. So proteins for anti-aging is a good idea where you can use nanoformulation.
Yes.
That's a very good
Those
idea.
are one of the major, these are the
That there we
major,
can-
this is the major area, means I have been working on superoxide dismutase.
They're working very nicely for vaccine delivery.
Yeah.
Because we usually give it in the inner part of the square, this thing, subcutaneous region. So at that time, it will just go and deposit there and keep on delivering the antigen inside the body slowly, slowly.
So I'll be needing your guidance in
issues,
doing
but
that
have a
because
little
I am not,
No
I
issues.
only work on enzyme part.
No
I
issues
need
at all.
collaboration for that.
No issues at all. No
Thank you
issues.
very much.
Whatever little. Thank you so much.
After a very extensive and highly energetic talk, I mean, I have rarely heard such energetic talks. Normally, we hear interesting, but at a very slow pace talks. So it was, it was pretty simple, as well as it covered things in depth. So congratulations, ma'am. And that speaks volumes about your scholarship. And we are really fortunate to have you here in Amity. Now I would like to introduce okay, sorry.
So may now request Professor Dr. Seema Bhatnagar, Professor Dr. Nidhi Chaudhary to please come forward and felicitate Professor Dr. Zina Thikpal, ma'am.
और confusion है
थोड़ा सा उसमें ना अनपब्लिश डेटा है हमारा. About the meeting at 2:15 BOS, you know. I told Deepika already. I would have loved to be with you. I'll come again, inshallah. BOS का आप समझते हो ना? Meeting है सवा 2 बजे. But that's problem. No, I just. Okay. So recycle bring down. Recycle. हमारा पेन ड्राइव भी था ना. बस इस आपको दे देता. अच्छा उधर है. कोई नहीं उधर होगा. Thank you so much. My pen drive is there? No, I just gave it. अच्छा ठीक है वहीं होगा. Thank you so much. Hello, thank you so much.
I will just. It's my privilege to introduce Dr. Himanshu Bavecha, a senior consultant, clinical pharmacology and clinical research at Yashoda Medicity. He is an expert in clinical pharmacology and clinical research. He has monitored and successfully supervised 150 plus US FDA approved clinical trials across a wide range of therapeutic areas, including oncology, endocrinology, psychiatry, critical care, infectious diseases, andrology, ophthalmology. He is also a research expert in Central Council for Research in Ayurvedic Sciences, Delhi. A big round of applause for him. And now I request him to deliver his keynote address.
Thank you. Good afternoon, everyone. I know I'm standing between you and your lunch, but don't worry. My talk will be to the point, brief and specific. So thank you very much, Dr. Tanveer, for your kind words, and Dr. Ashish. And I'm also thankful to the management, Dr. Seema, for inviting me to share my views on the regulatory of the drugs related to the drug trials in this international conference. So as my topic is reimaging the regulatory strategy from compliance to the competitive edge. So in this, we will discuss about what is the role of regulatory, how we conduct the regulatory compliance, what are the regulatory agencies, all that things. So this is my beautiful hospital, Yashoda Medicity, based at Indrapuram, Ghaziabad. And this is our top management. And now this is the agenda of today's session, that what is the regulatory affairs, what are the global regulatory agencies, what is the regulatory life cycle overview, what are the key submissions, documents, regulatory strategy fundamentals, and challenges and future trends. So previous speaker already set the grounds for me to discuss about what are the drugs, what are the drug trials. For conducting a drug trial, we have to take a regulatory approval. And every country has, as you already know, every country has a different regulatory agency. So in this, we will discuss about what are the different regulatory agencies and what are the different regulatory documents which need to be submitted during the regulatory submission of the particular drugs, particular new devices, and any another diagnostics, devices, drugs, everything related to the regulatory. So this will be the interactive session so that I don't want anybody will sleep. So regulatory affairs is basically a professional discipline. And I will also discuss for the students what are the scopes of regulatory. So this will be the interesting presentation for students also. And in the last, I will also give the take-home message to all the students, especially the PhD students, because after PhD, you have to enter into the industry as a professional and maybe in the regulatory affairs. So regulatory affairs is a good field. Why good field? Because it will give you growth financially as well as professionally, both ways. So regulatory affairs give you all the legal, what are the scientific, what are the administrative requirements which are set by the government authorities before and after the drug reach the patients. So any new drug, any new device, any new diagnostic, whenever they will have to undergo a clinical trial, that is a phase 1, phase 2, phase 3, phase 4. There are several regulatory applications, there are several regulatory requirements which every regulatory authority have to fulfill. And every pharmaceutical company, every biotechnological company have a regulatory affairs department to deal, or you can say to follow up, to take the regulatory approval from that particular regulatory agency. So these are the main regulatory agencies of across the world. FDA, as you all know, of the US, that is Food and Drug Administration. EMA is the European Medicine Agency. MHRA of UK, that is Medicine and the Healthcare Regulatory Agency. CDSCO, that is a Central Drug Standard Control Organization. So these all agencies set the standards, review the evidence, and grant the authorization for medical products to protect the public health. Why I'm saying to protect the public health? Anyone? Any guess? Exactly. So that wrong drug, wrong device will not reach to the public. For example, I will discuss about the Yashoda or any corporate hospital. Every corporate hospital have two committees to pass the clinical trial. One is Scientific Review Committee, that is SRC, and second is Institutional Ethics Committee. At MIT also, I just discussed with Dr. Seema, you also have an Institutional Ethics Committee, but that Institutional Ethics Committee will review academic projects that is related to the PhD thesis. But at our hospital, we normally prefer or normally we, you can say we address the clinical trials, hardcore clinical trials of various specialties of oncology, of endocrinology, of critical care, of respiratory, of new drugs, new devices, new diagnostics. So all that new particular devices, drugs have to undergo first of all a review of scientific review committee. The purpose of scientific review committee is to see that that particular study is scientifically sound or not. Is it scientifically valid or not? And institutional review committee's purpose is to see that that particular trial is ethically correct, or as one of my colleague already just raised the point, that is it not wrong? Is it not unethical? That particular study is not unethical so that we can protect the public health. So that two committees are very essential, very important, and that are already, as per for your knowledge, these committees are registered by the CDSCO and DHR. DHR is Department of Health Research. So these both committees are formed as per the ICMR guidelines. They are formed as per the ICMR guidelines. The chairperson is from the outside the institute, and all the members are from the outside the institute, so that the committee work independently and the committee will work unbiased. So this is the importance, this is the main purpose of these two committees at the corporate hospitals. Now, regulatory agency throughout the product life cycle. As the previous speaker already discussed about the product life cycle, so I will elaborate about what are the different product life cycle and how we will elaborate. First is discovery and the preclinical. What is discovery and preclinical? During the discovery phase or preclinical phase, it will be done on the animals. And during this, we will have to see that is this drug is safe to administer to the human beings or not. I'm just talking about the new drugs. New drugs means new molecules or the existing molecule which are indicated for new indication. Okay. So this is for the discovery and preclinical. Second is clinical development. Clinical development is all about phase 1, phase 2, phase 3 and phase 4. In India, normally the drug companies don't do phase 1. Phase 1 is not allowed in India. Normally in India, we do phase 2, phase 3 and phase 4. Phase 4 trial, also called as PMS, that is a post-marketing surveillance study. In that, we have to submit the IND, that is investigational new drug application, and the CTA, that is a clinical trial application filing. Protocol review, as I already mentioned, that protocol review is done by the regulatory agency also, and also done by internal that particular site. Site is basically that particular hospital where the trial have to be conducted. That review is done by whom? कौन करेगा review? अभी डिस्कस किया था. Speak little louder. CDSCO तो रेगुलेटरी एजेंसी हो गई इंडिया की, वो बाहर से रिव्यू करेगी. फॉर एग्जाम्पल कोई न्यू ड्रग ट्रायल आया है हमारे पास, ये शोधा में आया है, तो इंटरनली कौन रिव्यू करेगा? SRC and then institutional review this committee. Okay, so first of all we will submit the project, submit the protocol to the SRC, that is a scientific review committee, to see that that particular protocol is scientifically sound or not. And then after the SRC approval we will move to the IEC, that is a institutional ethics committee. And after the approval of institutional ethics committee, the ethics committee will issue the approval letter to the principal investigator to conduct that particular trial or to recruit that patient. Patient recruitment is main step in the clinical trials. Trials किस पे होंगे? Patients पे होंगे ना? So normally patients of that particular specialty, जिस particular specialty का वो trial है. Is it clear? Clear or not? चल ठीक है. So phase 1 to phase 3 oversight, this I already mentioned. Submission and review is the third step, that is the NDA, that is a new drug application, that is a MMA, that is a marketing authorization application, and BLA is biologics license application. The drugs are basically a chemical components, but we also have sometimes biologics. Biologics normally used in the chemotherapy, in the oncology department. So sometimes we also have to review the protocols based on the biologics. Biologics are used in the, as I already mentioned, in the onco department. So for that, after the phase 3, these 3 things are submitted as per the criteria, that is a new drug application, marketing authorization application, or BLA for the phase 4. Phase 4, as I already mentioned, is a post-marketing surveillance. In that, after the phase 3, the drug is exposed to the small real life evidence, to the real patients, to the group of patients which are admitted in the hospital or which are come for the hospital for the treatment in the daycare or maybe in the OPD. So this is a compilation. CTD is basically a common technical document. Common technical document compilation is there, which is submitted to the regulatory agency by that particular company, by that particular sponsor company or the CRO. There is a difference between the sponsor and the CRO. Sponsor company is that pharmaceutical company who basically want to conduct that particular trial in that particular company. For example, X is somebody, either my friend, who has a UK-based pharmaceutical company, comes to India and gives the contract to me as a CRO to conduct that particular clinical trial. So myself is a CRO and that particular my friend is a sponsor company. So there is a difference between the sponsor and the CRO. The sponsor will give some amount of money, some amount of financials to the CRO to conduct that particular clinical trial in that particular country. Why they are coming to that particular country and why they are contacted CRO? Because they are based in UK and US. They don't have any contacts. They don't have any regulatory knowledge. They don't have any, you can say, hospital sites, licensing. So all that things will be done by the CRO. That is a clinical research organization to conduct that particular trial. And most important, as per my slides, the regulatory approval will be taken by the CRO, clinical research organization, to conduct that particular clinical trial in that particular country. So that CRO plays a very, very important role in conducting that particular clinical trial. So in that way, the regulatory affairs is a very, very important and crucial department in that particular company. The regulatory affairs department is there in the pharmaceutical company, is also in the biotechnological company, is also in the clinical research organization, that is a CRO. Okay? So there is a huge scope of regulatory affairs department in every field which I am talking about. So fourth is the approval and the launch. After the conduct of the trial, after everything, there is a labeling approval. The labeling approval is the approval of the label. Every medicine has a label. So labeling approval is there, risk management is there, marketing authorization is there. And after that, post-marketing surveillance is there. Post-market surveillance is after the drug is launched to the public, we will see that the drug have a post-market surveillance, that we as a monitoring of that particular drug. So this is a comparison, agency comparison at the glance. I already discussed that there are 4 agencies. They are their established jurisdiction, all that things you can just read that there is a review time of 6 to 12 months, and the review time of the European Union is 210 days, and in MHRA it is 150 to 210 days, and in India you can see there is a long period of 12 to 18 months. So India will take a longest time to approve the drug. So these are the differences, and these are the key regulatory submissions and the documents. So these are the regulatory submissions and the documents which have to be submitted during the for the approval. That is the IND, investigation new drug, as I already mentioned, is submitted before the conducting of the trial. NDA is a new drug application. MMA, that is a marketing authorization application. IMPD, that is investigation medicinal product dossier. Dossier is normally submitted by the CRO or by the sponsor to that particular site, hospital site, for the members to review that particular study. In dossier, we have everything related to the study. That is a protocol, CRF, that is a case record form, informed consent, insurance of that particular study, and investigation brochure, labeling, everything, and all the previous data, that is the preclinical data and the clinical data, everything is there in that particular dossier, which is submitted and the also submitted to the regulatory agency and also submitted to the particular hospital site. Clinical trial authorization, that is CTA, is also submitted. Biologics license application, that is also submitted in the case of biologics. CTD, that is a common technical document, which is ICH Global, which is internationally certified and internationally accepted document. And it is 510K, that is a pre-market notification, that is basically in the agency of FDA. This is basically a market device clearance if the substantially equivalent to the predict. So it is basically a pre-market notification which is given by the to the regulatory agency that we are moving to the market notification for that particular product. So what are the regulatory strategy fundamentals? Till now, any question, any doubt, I am happy to answer. Yeah, please. No, not that way. But basically, point is that every country has a different regulatory agency. So China has also different regulatory agency, and their working style is basically India and China are the world's hub of conducting a clinical trials. Why? Because India and China both have a English-speaking population. Secondly, population-wise, we have a huge population, both India and China, and they both are basically promoting a clinical trials, because every developed country, whoever want to conduct a clinical trial and want to market that particular drug, these are huge markets, sir. Because every country, every developed country, for example, a company which is based in US or UK or any developed country you which name about, want to conduct a clinical trial in India or China. Why? Because they will get easily the population. The trial cost is cheaper as compared to the developed country because manpower is cheaper. Regulatory agencies are more or less similar as of India. They will also take the similar time as we take. And but their population and our population both have, so they will easily get the patients of that particular disease. And after the clinical trial, it will be easy for them launch that particular product after the patent. So they will get the patent that they conduct that particular trial in that particular specific country's population, and for 15 years their market is theirs. So they will compete in that particular country with the another pharmaceutical companies. So this is the regulatory strategy. Regulatory strategy is a proactive, forward-looking plan that defines how the product will never navigate a regulatory pathway from early development to the approval and the post-market management across one or more markets. So this is a target product profile, pathway selection, risk management, and evidence generation. So target product profile is basically a targeting a population with that particular product, with that particular indication, that in which we want to conduct that particular trial. Pathway selection is the approval, or you can say another things which are regulatory authorities which will give the approval to conduct that particular trial. Risk assessment. Risk assessment is basically a because every drug has a risk and benefit ratio associated with them. So risk assessment is very, very important. And then after that, the evidence generation is also very important, because after the conducting or completion of the trial, the evidence is generated that that particular drug is efficacious, or safety percentage is this much in that particular population. The side effects or adverse effects are this much. So all that evidence is generated after conducting a particular clinical trial in that particular population. So this is the different phases of the clinical trial, as I already mentioned. So I will not go in this much detail for this. So labeling strategy. So labeling is basically done for every drug, for every injection, for every dosage form, for every tablet, syrup, everything. In labeling, we will mention indication and the usage. Indication, that particular drug is used for which indication. Dosage of that particular drug, that particular dosage is, for example, paracetamol, is dose is 500 mg TDS. So that is the standard dose which you have to know, and every drug has a standard dosage of that particular drug. What are the dosage and administration of that particular drug? Contraindication. Every drug has a contraindication, that that particular A drug cannot be given by the B drug. So there are different drug-drug interactions are there. So that drug-drug interaction is a separate science, separate topic, which we can discuss, which we will discuss later on. But there are contraindications of every drug, and that the physician knows while prescribing the drug to that particular patient, that these are the contraindication while prescribing the patient drug. Warnings and precautions are there, adverse events, adverse reactions, and clinical pharmacology. As I already mentioned in my introductory point, that I am a clinical pharmacologist by profession. So clinical pharmacology is basically a science in the pharmaceutical field, in a hospital field. In the clinical pharmacology, normally we do the prescription audit. What is prescription audit? Anyone? Prescription audit is basically done by a clinical pharmacologist in the hospital, in which we see that the drug which are prescribed by the doctor to that particular patient is right or wrong. Is there any error related to that particular prescription? That are prescription error, that we call it as a prescription error. What are prescription errors? That doctor mentioned the right dose, right dosage form, right frequency, right indication. Is there any drug-drug interaction is there? If antibiotic is mentioned, then antibiotic is given empirically, or is any test is done, or if any test is blood culture or urine culture report is there, then the antibiotic is given as per the culture report or not. So these are the tasks, or you can these are the activities which are done or which are monitored by the clinical pharmacology department. Not for the doctors, but for the nurses also, because there are a lot of administration errors also, lot of dispensing errors also. I'm not scared you that there are a lot of errors in the hospital, but this is a, you can say, a level of standard of care which the nowadays corporate hospital is following, because we are going for NABH and JCI. These are the accreditation bodies, and they will demand that there should be a clinical pharmacology department which is headed by a clinical pharmacologist. So I have a team of clinical pharmacists under me, and we also have a clinical research department in which we will monitor the adverse drug reactions while the conducting a clinical trials. So there is a SI reporting also in while conducting a clinical trial. So both ways, clinical pharmacology and clinical research departments are interlinked. In clinical research, we normally monitor the SAE or adverse event of the investigational drug. Investigational drugs means new drugs. But in the clinical pharmacology, We monitor the SAE or the adverse event of already established drugs. When I'm saying established drugs, the established drugs which are already present in the market, which are already used in the market, but occasionally that particular drug may cause any adverse drug reaction. Why? Because it depends on the drug-drug interaction, it depends on the condition of the patient, it depends on the genetic factor, it depends on n number of factors that are monitored by the clinical pharmacology, and we will do the causality assessment. Causality assessment is basically a root cause analysis of that particular adverse drug reaction. Now, this is the most interesting slide for the students. So, after PhD or maybe some master students are there, you can pursue a career in regulatory affairs. Regulatory affairs, as I already mentioned, is a good field. And as a fresher, you can be selected as a regulatory affairs associate or analyst. This is a designation which is given by the companies. After few years of experience, you will become a regulatory affairs specialist. After that, you will become a senior regulatory affairs manager. After that, director of regulatory affairs, that is after 10 to 15 years. And after 15 plus, you will become the vice president or the head of global regulatory affairs. So when you become the global regulatory affairs, then all the offices of that particular company, which are located across the world, you will become the vice president and the head of that particular company. So this is a, you can say, so in the government also, you can apply. Normally in the government, the regulatory affairs, normally CDSCO is launching a contractual jobs, sometimes the full-time or part-time jobs. So you can check the website of CDSCO for the government jobs. And drug inspector job and drug controller job is already there on the websites, you can check. So this is a takeaway points. The regulatory affairs is a crucial and the cross-functional discipline, and all the regulatory agencies have a scope of regulatory affairs department, because regulatory affairs department deals with all the approvals which are required for that particular drug, related to the clinical trial, related to the preclinical approval, related to everything. And every regulatory strategy, everything is managed by the manager level. So everything is managed very, very profoundly, very, very carefully, because you can't imagine that not a single mistake you can afford, because every document, every dossier should be correct, should be verified by your senior before submission to the regulatory agency. Because if one rejection is there, then it will take again review of 3 to 4 months, and that will kill that particular time, and the time is money in the corporate. I am very serious, and please note this point, time is money in the corporate. So time is very, very crucial. So you have to understand the importance of time, discipline, and integrity if you want to go, if you want to pursue the career in regulatory affairs. So thank you very much. In the last, I just want to give a small note for the students. Just upgrade yourself. Don't stick to the curriculum. I'm not against any, I'm not saying anything, but point is that upgrade yourself in the field so that you can excellent in your career. And I'm just giving you the take home messages. Upgrade in your artificial intelligence. Artificial intelligence is the next revolution in the industry. आप किसी भी फील्ड में जाना चाहते हैं, आर्टिफिशियल इंटेलिजेंस आपको वहां पे मिलेगी ही मिलेगा. ठीक है, so please try to complete the course in the artificial intelligence, either a part-time course or an online course which are available on the net. Please do practice on the Excel formulas and the PowerPoint presentations, and also do any specific course which is related to that particular domain. जहां भी जिस भी फील्ड में आप जाना चाहते हैं, फॉर एग्जांपल आप क्लिनिकल रिसर्च में आगे कैरियर परस्यू करना चाहते हैं, सो मास्टर्स पीएचडी के साथ साथ एक स्मॉल क्रैश कोर्स, ऑनलाइन कोर्स, प्लीज प्लान टू कंप्लीट दैट पर्टिकुलर कोर्स दैट विल गिव यू एन एज over the others to pursue that particular, to excellent in that particular career. So that is very, very important. So all the best to all of you and thank you very much. I think मैंने बहुत ज़्यादा बोर नहीं किया आप लोगों को. That's wonderful, ma'am. That's wonderful.
Part of their curriculum. That is a part of their curriculum and each one of them in some form or the other, they are doing as an elective or other form there.
That's wonderful because as I'm just telling as a my personal experience. So Dr. Seema से मेरी इस बारे में बात नहीं हुई थी अभी तक. So but I'm just saying as a my personal experience to as a senior to all my junior colleagues that artificial intelligence is there in everywhere, in pharma industry, in research industry, in pharmacovigilance industry, in regulatory industry, even in healthcare industry. So our hospital is also working in AI. And we also have a AI, madam se just discuss hi kar raha tha that we have a AI hard command center. And ICU is basically ICU of the secondary health center of government setup is controlled by our hospital. And we are working in very aggressively, we are working for the, you can say evolution or you can say a development of AI in another departments, especially in radiology, so that the reports will be generated in a very quick span of time because the number of patients will increase gradually in our hospital. So we want to give the patient satisfaction. So this is, as I already mentioned, time is money. So time is very, very important and crucial. So just, madam is here. So I am confident that madam will definitely guide all the students very well. So thank you. I will not take much time. Thank you very much.
Just, no, you please be seated.
Sorry.
विनोद जी थोड़ा साइड हो जाओ.
Hello.
Sir,
Yes.
if we have a pure molecule ready, which has given promising results in vitro and after animal studies as well, how much time it will take for these clinical trials and what is the process and the charges?
Okay. You want to discuss here? Okay, fine. No problem. I will explain. Normally, yeah, that depends on the molecule, first of all. What is the indication and what is the nature of the molecule? When I'm saying nature of the molecule, it's a biologics, it's a chemical molecule. And after in vitro, we have to go to the in vivo, that the phase 1, phase 2, phase 3, phase 4, as I already mentioned. So this is an academic research project or it's a industry oriented, you have a collaboration with industry, pharmaceutical?
Yes, collaboration is there that can be taken forward. Now I am seriously into industry.
Okay, fine, wonderful. So we will discuss, we have a ethics committee and SRC, as I already told you. So phase 1 will be completed, or you can say BAB study, that is bioequivalent and bioequivalent study will be completed by that pharmaceutical company. And after that, you will move to any hospital. I'm not come here to promote any my hospital. So generally you can approach any hospital. If they have a patient of that particular indication, then they will check for the feasibility. There's a site feasibility questionnaire is there. The pharmaceutical company knows each and every process about that. So there's a site feasibility questionnaire. The PI will tell, PI means principal investigator will tell that that particular study is feasible at our center or not. When I'm saying feasible, that number of patients for that particular drug is there. For example, I'm just explaining in little more. फॉर एग्जाम्पल आपके पास कोई मॉलिक्यूल है जो ऑन्को का मॉलिक्यूल है, ब्लड कैंसर का मॉलिक्यूल है. ब्लड कैंसर के हमारे पास पेशेंट्स ही महीने में 10 आते हैं, तो स्टडी को कंप्लीट होने में बहुत टाइम लग जाएगा. सिमिलरली अगर आपके पास कोई ऐसा मॉलिक्यूल है जिसके पास हमारे पास फॉर एग्जाम्पल डायबिटीज के पेशेंट है, डायबिटीज के हमारे पास 100 पेशेंट पूरे हफ्ते में आ जाते हैं, तो दिस इज ह्यूज फिजिबिलिटी. So as you understand, there's a feasibility we have to check that that particular study, that particular trial is feasible at that particular site or not. If the PI, principal investigator, says that that particular study is feasible, then the research department role comes into the play. We will you have to submit the dossier to that particular site, to the research department. Dossier means same as I already mentioned, protocol, CRF, institutional, basically insurance, and the informed consent in English and Hindi, both languages. And all that, there's a form which are submitted for the SRC, that is a scientific review committee. After SRC approval, we will move to the institutional ethics committee. This is the same process which every hospital follows, not my hospital, every hospital normally follows. There's a fees also, but for the academic trials, normally we will discuss that how we can, because for the academic thing, normally the hospitals will just sit and discuss. But there's a fees, there's a fees of ethics committee, good amount of fees for the taking the approval, because the members are from outside, they are the expert members, they come here, and there's an expense which is related to conduct that particular ethics committee. So after ethics committee approval, the committee gives the approval to that particular PI and also to that particular company to conduct that particular trial, that is to recruit the patients, that is patient recruitment will start, the trial will be started, that is the phase 2, phase 3 or phase 4, either which particular phase you want to conduct. So this is normally be called as a academic studies. This is normally labeled as a academic studies. You can say academic initiated studies. So there are different type of studies. Research projects are there which are sponsored by the pharma companies. We also have a ICMR funded projects in our hospital. There's an investigator initiated studies. Investigator initiated studies, for example, I am a doctor, I want to design some protocol and want to check that that particular research has some outcome or not. So I will submit that particular protocol, that was called as an investigator initiated study. And your study termed as a academic initiated study.
So does the molecule which has shown result in vitro level, can that be started with there?
That we have to discuss with the pharma companies. Yeah, that's why I'm saying the pharma companies have to produce the results of BA/B studies. BA, that is bioavailability, bioequivalence studies.
Okay.
So that will be done at the that particular pharma company have a setup. So after that,
I'll be discussing.
They
with
will come to us for phase 2, not us, as you feel feasible with any hospital.
I'll be in touch with you. Thank you. Thank you very much.
Sorry, I am again standing between you and your lunch.
Thank you, sir, for such Thank you, sir, for such insightful and thought-provoking session. With this, we conclude our morning session for the day. And now I would like to request, thank Professor Dr. Zeenat Iqbal and Dr. Himanshu for such enlightening lectures. With this, I would like to invite Professor Ashish Mani, Professor Dr. Nidhi Chaudhary, and Professor Dr. Madhumita Ghosh to felicitate Dr. Himanshu Baveja. May I please request to please come forward, and Professor Navid, sir. I would like to also request ma'am to please felicitate our session chair, Professor Tanveer Naveed. So. So with this, we will proceed to the lunch. So please, I invite everyone to join us for the lunch. Okay, see. We can